तो नहीं होता फिर ठीक है। तो Good morning, everyone. I think there are only two students so far in the class. Who is the second student? One is Sharvari. Who is the second? Oh, there are three students now at least. This is not good. Like, we're making lectures for you, and only three or four students are coming. 1, 2, 12 participants are there. Okay, time is less. I have to start now with the biochemistry of water. So basically, as we know, the water is around 70% in our body, and it's very essential before we study about any other biochemistry or any other molecules we start to take. So I should have start with the biochemistry of water itself rather than going for amino acids and other biomolecules. So let's start with the biochemistry of water. So water is actually a solvent of life. All organisms are made up of around 70 to 90% of water. Eukaryotics are 90% and your prokaryotics around 70%. Um, and no organism could live actually without water. If water is removed, if anything would be dehydrated, then nothing could exist. Even as we human being, sometimes we say that dehydration happened. What is dehydration? Less amount of water in our body. If that happens, our body is not working well. So we are given excess of water then later on in order to fulfill the loss of water. So all any chemical reactions that are happening in our body, it's happening because of the liquid water. Also, your proteins, these polar, non-polar proteins within our body, they are also being held with the help of water molecules. They are transporting from one place to another just because of the water molecules. So these water molecules are actually polar in nature. Yeah, these includes as a solvent, chemical reactant, and fairly maintains a constant temperature of any organism. So it's a yeah. They say drink 2 to 3 liters of water every day. Why they are saying that? To keep our body hydrated. Even nowadays, it's more thing is coming up like you put cucumber, mint, lemon, and make a detoxifying water. Get a big jar of water, and then you put sliced cucumber, sliced lemon to make it more acidic and more nutritious. Then all the nutrients of that will come into the water, and it's more healthy. So yeah, water has many several unique properties. It acts as a medium in which all cellular chemical reactions occurs, and being a polar molecule, it attracts the other polar molecules and act as a universal solvent in the cells. For example, we put salt in the water. The solute dissolve in separate sodium chloride ions because water molecules break the weak ionic bonds and surround each ion sphere of water molecule. So when we add NaCl, uh, so these sodium and chloride ions, they got separated because these water molecules break the ionic bond between the sodium and chloride, and each ion is in sphere of water molecules. Then this chloride will be with the ions of your, I will discuss these water molecules, and others also with the water molecules. So an aqueous solution which consists of solutes in water is essential for chemical reaction to occur. So here we can see in the water we added a salt. These are the plus minus charges of sodium and chloride. Sodium is plus, chloride is minus. They are the undissolved chloride, so they are not dissolved in the water. The ones who are dissolved, so what will happen? The water molecule will like the positive H, this O2 H ions will like make a sphere around it, and dissolve them. We'll break the bonds between them and start dissolving them. And the positive ions, for example, sodium here, and the negative ions is your oxygen, they will all make a sphere around it, and so on. And slowly you will see your water molecule and your salt both are in equilibrium phase, and everything is dissolved in very well, and each ion having this sphere of the your water molecule. So that's the chemistry behind the dissolution of NaCl with the water. So water molecules are also reactant in many chemical reactions. Example of these hydrolysis reactions shown at the top of the page. Splitting maltose, we will discuss this later, how this is happening. Um, so yeah, so this was we were discussing. We having this salt, this crystal, and this is like square lattice crystal formation, and each molecule is getting separated and making a sphere around it. Yeah, this is the H2O, and HH being the gray one, and O means the O red ball is the this negative one, and your yellow are the sodium and green are the chloride. So in this way they make these spheres slowly and slowly. So water contains polar covalent bonds because, why is it called polar? Because it has oxygen and hydrogen which do not share electrons equally. So they do not share, so that's why it's still one is positive, one is negative. So it's not a non-polar, it's a polar. So oxygen exerts a greater pull on the electrons and gains a negative charge. So oxygen has a tendency to gain electron from those other two hydrogen molecules, and that's why it remains negative, where hydrogen as a result has a positive charge because of that. So as a result the water molecule has a positive end, yeah, the hydrogen, because of hydrogen atoms, and oxygen is slightly negative. So this negative and positive both ends are present, so this forms a dipole moment, and that's why it's called as a polar covalent molecule. The reason behind water calling as a polar covalent molecule is because of presence of oxygen and hydrogen, where oxygen has a negative charge because it's like pulling the electrons, whereas the hydrogen has a positive charge because of that. So having these negative and positive ends, it's make a dipole moment and your water molecules become a polar. So here we can see from the, so yeah, that's what we are studying here. We are studying biochemistry, right? We are not studying molecular biology or biology anymore. So we are understanding the chemistry part of biology at the moment. So how things are developing within our body. So here we can see uh, this is the oxygen molecule with the 8 protons in the nucleus and 8 electrons outside the nucleus. So 1, 2, 3, 4, 5, 6, 7, and 8, and one electron from hydrogen, one electron from hydrogen, and one proton. So that's how they are sharing. So if it's this way of showing it, 2, 2, 3, 4, 5, 6, 7, 8, and so on. So in at the end, the oxygen molecule will have your negative charge, and your hydrogen and hydrogen will have tend to have a positive charge. So we can see that oxygen is highly electronegative, whereas the hydrogen bonds are donor and acceptor. So high boiling point, melting point, heat of vaporization, and surface tension because of that reason. So oxygen is like want to have electrons from them, and from here too, that's why it's 2 delta negative, whereas this charge will be your delta positive of your hydrogen. So this is the bond polarities. There is one oxygen, one hydrogen, hydrogen, oxygen. So it has a charge to absorb these hydrogen positive ions. So it's make 2 delta negative, delta positive, and delta positive, right? So net dipole moment would be in this case on the top like this way. On other case with NH3, so we have 3 hydrogens, yeah, and there is 3 delta negative then and 3 delta positive. So bond polarities net dipole moment will be on the top, and there is a bond polarities, that is there is C in the center, oxygen on the right. So delta negative and delta negative, and in the center it will be the 2 delta positive, because these were like these negative and negative and negative, so they were moving towards up, so the net has to go up. In this case also up, but in this case both are making these charges on the opposite side, so that's why there will be no net dipole moment in this case in your CO2, carbon dioxide. In ammonia it will be, and in oxygen, in water molecule, sorry, it will be also. Further, as we have discussed so far, the polarity, net dipole moment, how salt is dissolved in it, so why it has high boiling point temperature and pressure, because of the bonding of the H2O. That's the main chemistry behind your H2O molecule. Where are you, students? You are here. That's the main thing behind it. Okay. So further, within the water molecule, it's also exists in 3 states. That is liquid, solid, and gas. Liquid, that is an aquatic environment outside us and inside us. Solid as an ice, and gas as a steam. Beyond that, they have surface tension. The cell membranes are moist protection. Capillary action in the plant roots, attraction to glass meniscus, and cohesion, water molecules stick to each other, held together by the hydrogen bonds. Whereas the adhesion, water molecules stick to unlike surfaces such as glass, plastic, due to the hydrogen bond between water and other polar molecules. So you will see if you put them on the glass or any plastic, so these water molecules do not stick to them. They readily get adhere from them, and they want to get out of there because they can't make these hydrogen bonds with these materials. But whereas the water molecules, they love to be in bonding with each other. If you put one droplet with another, they tend to make a big droplet with each other. And our cell membranes, they are also moist, and they are being protected by the water only. And also the capillary action, that is your water, your nutrients moving from roots to up part of your plants, leaves, and all those. So this could be possible because of the surface tension of the water property. And they have very high specific heat, so water can absorb or release large amounts of heat energy with little temperature change. So water stabilizes temperature in cells and ecosystem also. So that's why, as we can see, if just, I mean, whatever students we are studying here, don't think that we are studying, what do you say, some subjective things here. Try to correlate with your daily life, right? Like yesterday, we were discussing about amino acids, so various properties of amino acids. We have discussed around 20 amino acids, and they were essential and non-essential amino acids. And we found that the non-essential ones we do not require from outside, the essential ones we required, because non-essential we can produce from our body. So try to find out the all food sources. Yeah, make a list actually. Yeah, these ones. So try to make a list of food items from where you could get these essential amino acids, which are really required from outside diet. Yeah, but don't take these amino acids artificially, in the form of tablets. So some people are even taking them for the creating muscles and all. This is crap. Do not do that anytime. Be natural. Eat everything from these natural resources: tryptophan, valine, threonine, phenylalanine, methionine, lysine, leucine, isoleucine. So all these are possibles. You just have to be careful at what you are eating. So we have discussed many, like these amino acids has many functions in our body. So try to correlate. And I have discovered, we have discovered that how to these valines having different properties, cysteines, proline, phenylalanine. Just try to consume them in your day-to-day life, yeah, in day-to-day food life. The same way as we can see this water, that how important this water is, how it keeps our cells hydrated, how it maintains the ongoing life of our body very well. So try to correlate. So that's why the course subject is medicinal biochemistry. We should not we are not here just to discuss about the biochemistry of these molecules, of biomolecules that are present in our body, but how we can correlate in order to make our health healthy and better. Yeah. So in the morning, the best thing, the very best thing, that is that some people from the Ayurvedic perspective, I am a actually great fan of Ayurveda. Um, our ancestral, I have some books also related to that. So I'm even though I'm a PhD in uh, molecular medicine. I am a allopathic. I studied everything allopathic, but when I studied my Indian culture, our Indian culture, and found out that our great saints in the past, they have found very healthy things which have no side effects. Yeah, without much knowing, they are know about our how the body works, but not deep as we are knowing at the moment. So, but still somehow they found the cure by using these herbs and all those. So just put maybe cardamom or what is this called? This big one, cinnamon sticks. Yeah, put cinnamon sticks in the water, boil it, and or put some cumin seeds or put some turmeric powder. I don't know, whichever you feel like the best. Boil it. Drink 1 glass in the morning. It will clear your bowel movements and make your stomach acidic, and you're good to go for the whole day. And then fill at least 2 water bottles nearby your study table. If you're studying at the moment or you're going to the college, at least carry 2 water bottles in this scorching heat. You should never be dehydrated in this time. And these water molecules have high heat of vaporizations. So many, as we said, there are a lot of hydrogen bonds are made between each water molecule. So it's very, they have to be broken first before the water could evaporate. And the ice has very low density, so that's why they're floating. So water molecules in ice crystal are further apart because of hydrogen bonding. So when the ice is made, ice is actually is not, the bonds are very close to each other. It's just the water molecules become very far from each other. So that's why the density is low. This we have discussed, that it's a polar nature, surrounds ions, other polar molecules. So that's why it's very helpful in the transport of nutrients in organisms, for like plasma, cytoplasm, xylem, everywhere, in plants, in human, both. And it's a medium for chemical reactions, so hydration, dehydration reactions. So how is the water acting as a universal solvent? So most biological activities that are occurring, they are occurring in the aqueous nature, in aqueous solution, water-based solution. Water is able to dissolve small non-polar covalent molecules, ionic compounds, and other polar covalent molecules. So water-soluble molecules are described as hydrophilic, that is water-loving, whereas the hydrophilic molecules dissolve in the water. So all like there's philic and phobic, might be aware. So all those are able to dissolve them. So which are not able to dissolve in the water, they will be called hydrophobic, that is water hating or fearing, and they will repel water. And there's amphipathic. So there are three terms that are coming up. So hydrophilic, hydrophobic, and amphipathic. So amphipathic uh, is the one which having both hydrophilic and hydrophobic properties. So they're having these both water heating and water loving properties, where the hydrophobic have only heating properties, where the hydrophilic has only water loving properties. So lipids are actually amphipathetic. Amphipathic, yeah, not pathetic. Amphipathic. Why? Because they have polar head and non-polar head both, and one is water loving and one is water repelling. So both, and outside is the water loving, inside is the water repelling. So that's why lipids could be present outside with the water, but inside not. So mixture of one or more substances is called solutes, and dispersed in the dissolving medium called solvent. So the liquid medium will be called here is a solvent, and the solute would be the your solid particles here. So the sodium has the electron valency as 11, so 11 protons are present. Chlorine has 17, right? So towards hydrogen, your chlorine will be attached, and towards oxygen, your sodium will be attached. And positive ions are known as cations, and the negative ions are known as anions. Yeah, positive are the cations, and these are the anions. And this I was saying that non-polar substances are insoluble in water. So like these lipids are amphipathic. So they have polar head, which is a hydrophilic, and they have this non-polar tail, which is hydrophobic. So what is happening in this? This is the monolayer of these lipids. So outside, your all water molecules could be attached. Yeah. So this is the globular lipids we can see. Then inside, it's not possible. So because of this, it's the formation structure that is made in the case of lipids. It will be called as micelle. Yeah, micelle formation. In the class 12th, we used to study this micelle formation of lipids. This is the carbohydrates, and I still remember those time. So yeah, micelles formation will happen. And also, as we have discussed, it's very hard for water molecule to dissolve non-polar compounds, but sometimes it does. It dissolve. Yeah, it's the same thing is coming in different forms. That water molecule could dissolve these chloride ions and able to dissolve the water and NaCl. Same thing is there also again. Your all positive hydrogen ions will cover your chlorine ions. This is shown in a better systematic infrastructure here. And here the sodium ions will have all the negative ions covering by it. So in this way, your salt is being dissolved very well in the water. And there's a solvation and hydration shells. So depending on the pH, how is the pH, what kind of micromolecules that you are dissolving, such as proteins, which might contain charged groups, will have some net charge, either positive or negative. So that's depend upon your dissolving of that thing in the water. Yeah. So we have discussed this amino acids. They having different alkyl groups. This R side chain. So according to that, so if whether they are polar, whether they are non-polar, whether they have positive charge, whether they have negative charge. According to that, and how is the pH afterwards. So that's they are depending upon. And also cells of your body and blood contain many polyelectrolytes. So molecules that contain multiple same charges like DNA and RNA. Yeah. So your DNA and RNA, they are actually negatively charged molecules. Why they are negatively charged? Because of the phosphate group present over the side chain. So this gives them net negative charge. And they are also called polyampholites that are in close proximity, having the both charges. So close association allows these molecules to attract through opposing charge groups. So the presence in cells and blood of numerous small charged ions like sodium, calcium, magnesium, manganese, potassium leads to interaction of the small ions with the largest microions. And this interaction can result in shielding of electrostatic charge of like charged molecules. When when like sodium, they all meet together, so they could make a big bunch of positive ions. So they could make a shield of that, and because of that like charged molecules, they could be electrostatically charged, giving a shielding effect in the water. Yeah. And then further ionization of water. So we know that H has these lone pairs of electron outside, these four. Another H2O has also that. So one oxygen could give this to the H and H give to the oxygen. So what will happen? H will be your positive because it has given its electron, and this OH will be the negative. O will be the negative because we are moving this H out of it. So what will happen? H2O plus H2O give rise to H3O. So yeah, this H is being broken up and coming up there. One more H, and what happened? This will be OH negative. So in this way, our water is ionized. So H2O plus H2O give rise to H3 plus OH negative, and also further H2O could make broken into H plus and OH negative ions. So as we know, the equilibrium constant is product upon substrate. Yeah, the products are now here H plus and OH negative ions, right? So we have H plus and OH negative ions, and so H plus OH negative in the product, and the substrate is your H2O, right? So let's say um, yeah, so K equivalent, they have calculated that the equilibrium constant is 1.8 into 10 raised to power minus 16 molar, and the H2O concentration here is given around to be 555.5 molar. So you take this H2 concentration and K equivalent to there, and you multiply them, and you get this value as 1 multiplied by 10 raised to power minus 14 m square. Yeah, this will be your water constant, H multiplied by OH negative. What if your H plus ions and OH negative ions, they are in equal amount, right? Then the value of this H plus ions would be 1 into 10 raised to power minus 7, half of it. So ionization of water releases hydrogen ions. Yeah, so whenever the ionization of water molecules happening, so you will remove uh, hydrogen ions and hydroxyl ions, OH negative. And pH scale, as we know, it ranges from 0 to 14, and the concentration of H plus ions is the expression of pH scale. Yeah, so pH scale, it ranges from 0 to 14, and the concentration of this H plus ions is known as to be the pH scale. And in principle, we define pH as a negative logarithm of the concentration of hydrogen ions minus log of H plus ions. How many hydrogens ion are present? So typically, mathematically, we represent as minus log of H plus ions. Yeah. So if pH is 6, that means it's 0.000001 moles of hydrogen ions per solution, and so on. What is this? So if you want to go for some calculations, some problems, you can, you're most welcome to do it here. Some water related calculations. So as we know, within the cell there is acid and base, and this is also is happening because of the hydrogen and oxygen OH ions, and they could be maintained because of the water. Yeah, because of the water. So cell chemistry is sensitive to pH changes. So an acid is a chemical substance that donates hydrogen ions to solution, and base is the accept. So mainly the acids are the one which donates, and the base are the one which accept. So let's say if this is the greens are the here on the top are acids, so they tend to, in the water molecule, they tend to lose these hydrogen ions, so they are acidic in nature, whereas the base are the one which accept these H plus ions. So both acidic and basic at the moment. So acid, as we discussed, donate, and base is the one which accept. So acid donates hydrogen ions to solution, while um, bases, they remove hydrogen ions from the solution. The bases tends to remove, acids tends to donate. So pH scale indicates the number of hydrogen ions solution and denotes the relative acidity of solution. So how many iodine hydrogen ions are present within your solution, that is a minus log of H plus ions, is the pH of your indicative solution. And in order to maintain our cell like on the longer level, whether it's our liver, lungs, stomach, heart, kidney, any cells of our organ in our body, prostate, ovaries, so the pH level has to be maintained in them. Yeah, if the pH get more basic or more acidic, it's very harmful for the cells, and cells might not able to survive that. So for the same, in order to maintain that, water is again playing a quite essential role to maintain the acidity and basicity of the cells. So these cells are very sensitive to pH changes. So in aqueous solution, most of the water molecules remain intact, but some can dissociate into the hydrogen ions and your OH negative ions to rapidly combine in the reversible reactions. So like H2O could be having this reversible reaction of H plus and OH negative ions. So these H plus and OH negative ions need to be in proper amount. They should not go up and down. So besides water, other compounds in cell can release H plus OH negative ions, which affect the cell function. So for our purpose, an acid is a chemical that donates H plus ions to water or another solution. So by contrast, a base is a substance that accepts hydrogen ion solution, often by combining them with the hydroxyl of the base. Okay, not too much. So yeah, this one. So acid and base must be balanced in cells. They need to be balanced. So an acid is a chemical substance that donate H plus as a base accept. So pH scale here we can see shows the acidity or alkalinity. There is called alkalinity and also basicity. So acidity is also known as alkalinity. So here we can see that, oops, pH 0 and this is pH 14, right? This is the where the pH 1 to 3 is the range for the acidophilic microbes, where the acidic microbes could survive, and this is the range of your fungi from 5.5 to 8.5, and also the 6 to 8.5 is the range of where most microbes are growing. So this is around the neutral pH. Your fungi and most of the microbes are growing, and in this extremely acidic, your acidophiles microbes are growing. And let's see how the pH of various things are. So concentrated hydrophilic acid, yeah, concentrated HCl, it has the extreme 0. The stomach acid in our body 1, lemon juice only 2, yeah, vinegar, wine, pickles around 3.2, tomato juice 4.2, very acidic, black coffee very acidic 5, saliva, our mouth saliva. So they tend to say, as from saliva, I remember, if you have any dog as a pet in your house, or if you see on the street dogs, what they do tend to do in the early morning when they wake up, or mostly in the house, they tend to lick their feet or wherever they have, you know, mm, they have any wounds over the leg, over anything. They tend to just keep licking it. So it's actually their animal instinct that they're doing. So animal instinct or even human instinct, it says that our saliva has healing properties. Yeah. So our saliva has healing properties. So any dog or even tiger, if a tiger sees that his neighbor lion or tiger is injured, so they tend to lick each other to that wound, and it will heal actually with the time, because saliva has healing properties. So in the morning, it also says that do not just do brush straightforwardly. The saliva that we have in the mouth, we should gulp it with the help of with a glass of water. and it's actually healthy for our body. I don't know how much truth is behind that, but I was told by one of my elder people. So I'm doing it. It's helpful. It's not bad. And but yes, as a human beings, we are civilized now. We have band-aids, we have creams. But earlier time, there were people, they were licking, they putting their saliva over the wound and let it heal, and it was working actually. This is something about saliva that might be not aware of. Then milk is also around this neutral pH 6.2, and the pure water that we are drinking, it's of pH 7. Yeah. And blood is also neutral pH. Then baking soda, ocean water, they are now coming onto the basic, more on the basic side. Commercial bleach, yeah, commercial ammonia, oven cleaner, and sodium hydroxide, NaOH, concentrated, 14, extremely basic. So this table actually gives us an idea. So what is where what and how things are working within our body. Yeah. So acids are hydrogen donors. So here we can see carboxylic acids giving rise to COH, CO negative H plus ions, and the bases are the acceptors NH2. They accept NH3 plus ions. There are a lot of H plus ions here. Then it will have a very low pH. And when there is, because um, yeah, it will be more basic. And then if it is lots of OH negative ions, that will be very high pH. So this is sorry, weak acid and bases equilibria. So strong acids and bases, they dissociate completely, whereas the weak acids, they dissociate only partially. So enzyme activity is, they are sensitive to pH. So weak acid base plays important role in the protein structure function. So this is our pepsin. So who's efficient? They are efficient at the more acidic level, so around 2, and the trypsin is around 6, and the lysosome is around 4.5. So it depends upon which kind of enzyme, the pH they are, they will be highly effective. And then acid conjugate base pairs, so Ha give rise H2O. Then this it will be A negative H3O plus ions. So at the end it will be A minus plus H plus. So Ha is your acids which donates H plus ions, that will be called Bronsted acid, and A negative is the conjugate base which accepts this H plus ions, that is Bronsted base. So we put this equilibrium constant here, um, H plus A minus divided by Ha, whereas K and pKa value describe the tendency to the loose H plus ions. If it is a large Ka value, then it's a strong acid. If it is small Ka value, it's a weaker acid. And if you put this pKa value, it will be equal to the minus log of Ka. Then carboxylic acids, which are organic acids also. An acid functional group in organic acid is the carboxylic group, and the carboxylic group is a protein donor here. So COH give rise to CO negative in the water solution. And there are organic bases which accept proton acceptors, gains a hydrogen, become positive charge in the water solution. So like NH2 becomes NH3 plus ions. And pH scale, as we have discussed, it's indicate the acidity and alkalinity of solution. So cell chemistry is very sensitive to changes in the pH itself. So the pH is the concentration of H plus ions and the negative log of the hydrogen ion concentration in an aqueous solution. So this pH scale was derived by Sorensen in 1902. So here we can see HCl, gastric contents, tomato juice, cow milk, human blood plasma, human pancreatic juice, ammonia, and sodium hydroxide. So they are just providing us various examples that we have just discussed. Oven cleaner, how everything is divided nicely. So this is the pH where acidic rain happens. So all fish will die. Yeah. And this is the frog, eggs, tadpoles, crayfish, mayflies die in this pH, and rainbow trout begins to die. All fishes that are present in the lakes with these different pH, things tends to get destroyed because of the uh, environmental conditions. This is the common pH meter that we see in the labs, and this is the strips that we use, hydron paper. So whatever the color that is showing after taking the clip, it will indicate that your solution is of that pH. But we tend to make solution in the lab, we tend to use this electronic pH meter. Yeah, I hope all is well at the end. Okay, so buffers are combination of weak acids and buff and base. So buffer prevents pH shift. So buffers are aqueous systems that resist changes in the pH when small amounts of strong acid or base are added. So a buffered system consists of weak acids and conjugate base. So they are very effective at the pH that are within +1, -1 pH unit of the pKa. So buffers are actually a mixture of weak acid and weak base that maintains the acidity and base balance in the cell. So excess hydrogen ions can be absorbed by the base and too few hydrogens can be provided by the acid. So they maintain your hydrogen ions concentration equally in the body with the help of buffers. So it is important for all organisms to maintain the acidity and bases in their cells because chemical reactions and organic compounds are very sensitive to pH shift. So likewise, when most microbes grow in microbiological nutrient medium, the waste products produced may lower the pH of the media, which could kill the organism. So this is the aqueous phase of blood passing through the capillaries in the lung. So HCO3 minus, H plus ions coming, H plus ions going, it making H2CO3, and then again H2O is moving, H2O is coming in. If it is going, then it's become your carbon dioxide. So this is aqueous liquid in the blood cells, but in the lungs it become a gaseous, and it will keep going reversible also. So by addition of H2O, it will be H2CO3. By removal of H plus ions, it will be HCO3 negative. So buffer capacity, so amount of base added, so buffer range is around 4 to 6 normally. So buffers are combinations of weak acids and they prevent the pH shift normally. So the chemical reaction is happening. So if needs H plus ions, the potential pH increases. If it is excess pH H plus ions are produced, then these will be absorbed by the base, this acid base buffer system. Then H plus ion dissociate from the acids and they go further to the chemical reaction. So this buffer will maintain your H plus ions concentration properly in total. So, so that's it from the biochemistry part of our um, water. I hope it was a bit big lecture today, but I hope you had good understanding of it from the chemistry, core chemistry of biochemistry. So we'll share this link. So till that time, take care. We see us tomorrow with more about now. What do you say? Yeah, lipids. Something about lipids and how they're working from the medicinal biochemistry perspective. Yeah, till that time, take care. Have a nice day. Bye bye. Thank you so much.